Okay, good afternoon, folks. Thanks for sticking around. Today you're kind of getting a 3-for-1. We're going to talk about Carpophilus, Beetle, Leaffooted bug, and Navel Orangeworm, all in quick succession. So just trying to get to the most recent updates on research for each of these insects. So we'll start off with Carpophilus, since David just gave a great overview of that. So up here we've got Team Carpophilus, effectively, Jalinda Rajal, David Haviland, Sudin Gaiwali, and Raman Bansal. We have all been working together since we first encountered this insect back in the fall of 2023. So we had simultaneous finds of Carpophilus beetle infesting almonds and pistachios in Madera and Kings counties, respectively. We subsequently surveyed throughout the Central Valley, primarily in the San Joaquin, and were able to confirm that there were infestations in each county in the San Joaquin Valley. And at this point, we have found it on almonds and pistachios, as I mentioned, but also walnuts as well. So this insect, we know it can affect all three tree nuts. We've confirmed that here in California. And most recently, we had a find of infestation in almonds in Colusa County as well. So it is present in the Sacramento Valley. Now, what David was mentioning about how big of a problem is it, presence-absence is one issue, but the intensity of infestation is another. And I'll talk a little bit more about that here in a minute. As he mentioned, these things overwinter in the mummy nuts down on the orchard floor as larvae or adults or pupae, and then they can pupate in the soil. Those adults come out, and they'll, over the course of the season, continue to reproduce on those mummy nuts, but at whole split or whole slip in pistachios, they'll some fraction of those adults will move up into those new crop nuts. So y'all kind of got an overview of that already, but just to reiterate it a little bit. So monitoring, we currently don't have any traps or lures available for this insect. The best thing you can do is inspect directly either mummy or new crop nuts. So you're cracking out a bunch of nuts looking for signs of infestation. The adult will chew a hole into the shell of an almond. As mentioned, they can't get through that shell of a pistachio, and then deposit eggs onto that kernel. And so then those larvae will burrow or tunnel into the kernel itself. So what you end up seeing in an infested nut, if you can see that circular hole in the shell, that's great. Sometimes that's kind of hard to see. But also you'll typically see a lot of this kind of a fine white powdery substance. In pistachios, it's more of a green color. So that's this mixture of insect frass and chewed up kernel meat. And so typically we'll see a nut like that, blow that dust away, and then you'll see this oval-shaped tunnel in that kernel. And so that's a clear sign of the infestation. So monitoring is fairly limited. We'll talk about some research about that later. Management, biocontrol is very limited. Chemical controls are also very challenging, primarily due to coverage issues. Again, I'll get into some of the research around that here in a minute. So at the end of the day, crop sanitation is really the most critical practice for controlling this insect. And you saw on David's slides, those guys in Australia are using these hammer mills. You really have got to shred and destroy these mummies. So they like wet mummies down on the ground. This picture here on the bottom with that hole, this is some research from Australia where they actually buried infested mummies as far as 3 feet down, and they were able to survive, and the beetles come up to the surface. So you have got to destroy those things. So here's some of the research we've been doing in California initially to characterize the seasonal phenology of this insect. So what you're seeing here is some pictures of these emergence cages that we put into orchards. It's like a little tent, but it has no bottom to it. We filled those up with mummy nuts, and we've done this in almonds and pistachios and walnuts now. And then we go and can look at those mummies every week or every other week to see what sort of activity we're seeing over the course of the year. So we know they overwinter on the mummy nuts. When do they become active? When do they start reproducing on them? And then inset here, there's I think this little pointer will work there. You can see this little collection cup at the top of the tent. So any adults that are trying to actively move off of those mummy nuts will go up into the top and get caught in that vial. And so for us, that's a sign of them trying to move away from those mummy nuts, around whole split. So just some preliminary data. We have a huge backlog of samples we're processing through. These little tiny black beetles are difficult to identify, and there's a lot of other species that are very closely related that are not pests, but they're in the orchard, so it does take time for us to get through all of this. But the key thing we're showing here is that, they do become active on those mummy nuts in the in the early spring and those little rises, those little peaks and valleys there, that's multiple generations of this insect. So we think they have something like 6 to 7 generations per year just based on the weather data and some of the preliminary observations we've made. So they definitely become active on those mummies in the early spring. And then the second data set here, this is showing the adults that we captured in that little collecting vial at the top. And that's, this is data from an almond orchard. So this is occurring, more or less as we approach whole split. So there must be something in the volatiles from those split nuts that are attractive to those beetles, and so some fraction of those adults move up into the canopy. So phenology and temperature models, there's been some work already conducted in Australia to characterize the developmental biology of this insect on an artificial diet. So I've got the minimum and maximum temperature ranges there, and kind of in an ideal condition, around 86 degrees, it takes them about 4 weeks to complete a generation. So if you kind of do that math over the growing season, that's where I'm kind of looking at 6 to 7 generations per year. So they have multiple generations. They're building up that population over time. What we're doing now is looking to characterize their developmental biology in a similar way, but having them develop on almonds and pistachios and walnuts. So host quality, so the nutritional content of what they're developing on, can have an impact on their developmental rate. So we want to better understand that to just add some more nuance to our understanding. So evaluation of chemical controls. I've got a couple sets of slides here that were lent to me by colleagues. So the first set here, this is from Bob Van Steenwyk, and you can see a summary of these results also in the executive summaries in the little booklet today. So what he did initially has been to look at a series of lab assays. You're basically, applying these pesticides to the beetles in a petri dish or something like that, and then looking at the mortality. So this first slide, this is showing a number of different products that he's looked at so far when they applied it to the adults, some of them in green having pretty good efficacy, the yellow a little bit less so, and then the red the least effective. And then larvae, the similar set of, or the same set of compounds, and again, ranking them, as kind of, high, medium, and low in terms of their efficacy. So this is good work in that it's helping us narrow in on the active ingredients that might be the most effective, to take to the field. And in his group, that's their plan now, is they're putting together different spray programs that they want to test out in the field. As mentioned, the challenge is coverage. These things are tunneling into those nuts, and it's hard to get that compound in contact with the insect. And so Jerlinder Rajal, he's been leading the field trials for the chemical control efforts in our project, and this is a summary of his findings from the past 2 years. And so far, there's no statistical differences between any of the compounds that were tested, and there is some overlap here between the compounds that Bob was looking at. So we're going to continue working on this. This is not to say that chemical controls are out of the question, but they are very challenging, and as evidenced here with these field trials. So the pheromone lure, so there has been a pheromone compound identified by the Australians that they have been willing to share with us to conduct field trials in California. They are working with they're kind of looking for a commercial manufacturer to scale production of this. So this is a compound that's not available to the industry yet, but we're hoping that in a few years this will become available. So the way it works, you've got this little bucket trap that you put down at soil level in the row middles. It has the pheromone, and then there's a host plant volatile type of co-attractant. And in our testing to date, these things are very effective. So this is an image from just 1 week of sampling. There's like 20,000 beetles in that bucket, most of which are Carpophilus truncatus, the target species that is the pest. But there are some other species of beetle that are drawn into these traps. So we continue to work with our colleagues in Australia to refine the use of these traps, to test kind of different blends and rates of pheromone to optimize that trapping system. What this is showing is a breakdown of the different species of beetles in the genus Carpophilus that we have collected from these different orchards that we've been surveying across the Central Valley. And what I want to point out here is the red, that's Carpophilus truncatus. That's the one we really care about. And here's all these closely related species, none of which are pests in orchards. They're just consuming, decomposing stuff, not really causing a problem. but they're easily confused with Carpophilus truncatus. And what I'm trying to point out here is I was talking earlier about presence absence. Well, up in the North San Joaquin Valley in Gelidner's samples, you have about 50% of what he was finding is Carpophilus truncatus. And in contrast, David and Sudin up in the kind of southern San Joaquin and Sacramento Valley, respectively, they do have Carpophilus truncatus there, but it's just a small fraction of what we're finding overall. So we're going to continue to further characterize this and conduct these types of surveys, but just, again, sharing some preliminary findings. And then finally, there's a whole range of different, outreach and extension activities that we as a group have conducted over the past 2 years, all of which you can find through my lab website, through UCIPM, through Gelidner's website. So there's notably this pest ID guide that we put together that differentiates the signs of damage or infestation from navel orangeworm and ants and Carpophilus beetle. So there is, can be some similarities there. So this does a nice job of kind of contrasting all that, so you can differentiate and know what you're seeing in the field. We appreciate the support from the Almond Board as well as the California Pistachio Research Board and the USDA to support all of this work. We are going to continue our efforts to characterize the phenology of this insect in California, looking as well at regional intensity of infestations and continue to work on refining that pheromone lure. In terms of control strategies, the pesticide trials are ongoing. We've also got a study looking at the impacts of different soil conditions on their success in pupating in the soil. So the larvae drop down, they pupate in the soil. Different soil types might be more or less difficult for them to complete that stage of their life cycle. And then there is some early work in Australia looking at mass trapping with these pheromone lures. We're kind of waiting to see how that goes, and we'll make a decision about whether or not that might be a good strategy for California. So again, just want to acknowledge the funding that went into this project and all the lab assistants and cooperators that helped with a number of field and lab assays. Okay, that's insect number one. Leaf-footed bug. So this is a project we've been working on for a number of years with steady support from the Pistachio Board. It's a collaborative effort with myself, Kent Dana, another entomologist at the Kearney Ag Center, and then Jocelyn Miller, a chemical ecologist in our Department of Entomology down at UC Riverside. In tree nut orchards, you can find these 3 different species of leaf-footed bug, Leptoglossus zonatus, Clipialis, and occidentalis. But really, currently, about 95 to 99% of what we find is Leptoglossus zonatus. So all of these things characterized, they got that big kind of flared feature on their back leg. That's kind of the leaf-footed part of the name. And then zonatus has got these very prominent kind of white to yellow spots right behind the head. So that's how you can differentiate them. These are all pierce-suck feeders. They cause all sorts of damage. They're focused on the new crop developing nuts, and that feeding can impact the yield and the quality of the crop, and also facilitate the transmission of certain pathogens. So, bad things all around. Very difficult to monitor for. Right now, you've got to go out and beat sample the tree canopy or look for signs of damage, which is very time and labor intensive. So, as mentioned a number of years ago, we started to develop a passive trapping system based on pheromone compounds produced by the male leaf-footed bugs. So, I'm kind of summarizing in one slide over multiple years of work. But at the end of the day, there's this bouquet of pheromone that the males produce. It's composed of 10 different compounds, one of which was entirely unknown to science prior to this work. And so we were able to name it leptotriene, kind of after Leptoglossus. And that really is the most important compound. So we screened all these different individual compounds and in combination. And at the end of the day, leptotriene is really the heavy lifter there. And so that's what we've worked to figure out, not only how to synthesize, but efficiently synthesize it so that a commercial manufacturer could produce a lure at scale. And so the stage we're in now is evaluating a couple different iterations of lures that are being produced by commercial pheromone manufacturers. So this is a slide from our most recent trial in pomegranates. What you're seeing there is the UCR, that's our in-house produced version of leptotriene, and looking at the attraction of that relative to these lures that are being produced by AlphaSense as well as Trace. And both lures look great. We're super excited to see that. So there is a viable lure. And then the last one, that's just our hexane control. Once we had a lure in hand, we revisited our trap selection. If you recall, earlier presentations, we've really been doing a lot of this work using this hanging panel trap. It's kind of a very large thing. It's not easy to use. We really would like to get away from that. And so what we've recently been doing is looking at the performance of this black or yellow panel trap relative to some other trap types. And what I'm excited to share is that these yellow sticky traps and clear sticky traps, in this trial, these were produced by Trace. Alpha Sense does have their own version of a sticky trap. Now they've basically created an improved glue that's stronger that can hold on to these leaf-footed bugs. When we did assays 7 years ago with sticky traps, the leaf-footed bugs would basically walk right over the glue, or they'd get kind of stuck in it and rip their leg off and fly away. They just weren't very effective at capturing these insects. So we're really excited to see that these different types of sticky traps are having some ability to capture this insect, and that's a much easier to use type of trap. So here's what Alpha Sense is selling. You can look up this Lepzon on their website, and then Trace. They've got their version of the trap that they're selling with here, or their version of the lure they're selling with the sticky trap. Going back to Alpha Sense, they're marketing it with the hanging panel trap you can see there. Word of advice, we are still trying to figure out how to best use these traps. It's really exciting. We have a trap and a lure, and now, you put that on your farm and you get leaf-footed bug in it, and do you spray? Do you not spray? What are the populations like in the tree canopy? All of that stuff is now things that we're trying to figure out in our next phase of this research. And so just a number of different objectives that we're pursuing right now. So this is a male-produced, as mentioned, a male-produced aggregation pheromone. So we're trying to now understand, if a female is reproductively mature, if she's mature and mated, do they all respond differentially or the same to these lures or not? Again, just trying to understand what this is exactly pulling in. Most of what we capture in these traps so far are females, which makes sense. But again, just adding some more nuance to it. In parallel, we're also going to start developing an automated trap for leaf-footed bug based on this lure and a sticky trap. So we've been looking at delta traps with a camera with a sticky liner, and we've been able to pull leaf-footed bug into there. And so we're going to start working to develop an automated trap using that technology. You have similar smart traps right now available for navel orangeworm, so kind of following that model. We're also trying to use these traps to better understand the timing of these insects' activity in almond and pistachio orchards, and more importantly, almost the timing at which they move into those orchards. And we've started some of that work already for the past couple of years in almonds. We're going to continue that now in pistachios, and the idea is that we would be able to create a risk map based on the composition of the landscape around a given farm. So if you have the right habitat types that would support overwintering leaf-footed bug, or later in the season, populations of leaf-footed bug that are developing on other crops, they are not exclusive to tree nuts. You see them in veg crops. You see them in weedy vegetation, things like that. And we're doing some gut content analysis to better understand what they're eating and what time of year, but really trying to give folks a better idea of where they should prioritize their monitoring and management efforts for these insects. Dispersal capacity, so it's a big insect, big wings. We're trying to better understand how they fly. I tried to put a video, which is always very dangerous to do, but that right there is a little leaf-footed bug on our flight mill. They spin round and round. It's like a little treadmill for insects. And maybe they're going to get the video to work here. Maybe. Bummer. Well, and then I mentioned the gut content analysis also. So we're able to dissect their stomachs and see what crops and other plants they're actually feeding on. So all of that in combination will help us better understand where you have higher and lower risk of infestation. Oh, there it goes. Look at them go. They're just flying around and round and round on that thing. So again, just want to acknowledge the support from the California Pistachio Research Board and the Almond Board of California have been fundamental to driving all this work forward. And then, of course, dozens of lab assistants and collaborating growers and pest control advisors in this project over the years. Okay, so closing in, regional IPM for navel orangeworm. So we know that navel orangeworm has a strong flight capacity. And you can do everything right on your farm. You're sanitizing, you're doing really good monitoring, putting in mating disruption, you're timing your sprays, you're getting the crop out of the field in a timely manner. But all of that can be undermined by movement of these moths from adjacent orchards into your farm. And so this insect, I've talked about this before. We think this is a really good candidate for regional or area-wide management. Different words, but kind of the same idea. So we have a new project that we started last year to develop what it's called a decision aid system for navel orangeworm that's going to help facilitate this regional coordinated management. We've got our project team here, myself, Chuck Burks at USDA, Jalinda Rajal with UCIPM, Dave Crowder at Washington State University, and then Joe Coelho and Carly Clark at the American Pistachio Growers. So those are the main drivers here behind this project. We're funded initially by the Pistachio Board and Almond Board and appreciate their continued support, and we've been able to leverage that already into a larger grant from CDFA, which includes this larger management team here. If I can, you can see it there. So it's a combination of support from commodity boards, processors, and growers and PCAs. Definitely not an exclusive club. If you folks want to be involved in this, please come talk to us, and I'll talk about enrollment here in a minute. So what we're trying to do is set this up initially in Stanislaus County and West Fresno County. It's kind of where Jalinda and myself and Chuck are positioned with our work. And the idea is to set up a regional network to monitor navel orangeworm using smart traps. And the idea here is to kind of create almost like a weather forecast for navel orangeworm, so growers can see, what's going on their farm, what's going on in the region, how do their trap counts compare to what we're seeing in other parts of the county. So that's something that we're going to get set up this spring, and we'll be displaying all that information through this decision aid system website. So we'll be sending out announcements soon once the website goes live, but basically you'll be able to log in, set up an account. It's free of charge, and there's going to be this regional monitoring data, and then also you'll have the ability to run navel orangeworm as well as tree phenology models for your orchards using high-resolution weather data that is part of this platform. We're also working to integrate the CIMMYS weather network as well as the Wild Eye weather network into this, so you have different options. And participating growers are able to actually plug in their own weather stations as well to this, and if they want to reference that, of course, for running these models. There's also a series of updates and like I said, forecasting information that's going to come out for folks that are using who have accounts through this website. So that kind of covers the gist of that. And then there's kind of a more involved version where we actually will use this platform to facilitate the creation of these coordinated neighborhood groups, is what we're calling them. And so the idea is that a group of growers could band together and try to adopt mating disruption and other best management practices at scale across their region. And so there are some incentives we're putting together to facilitate that, but the idea is that to not just give you more information about what's going on with this insect in your region, but also create a way for people to better coordinate and adopt and scale best practices for navel orangeworm. So just breaking down who's doing what here, the decision aid system is something that was pioneered at Washington State University. Dave Crowder is running that and setting that up and developing these forecasting models. American Pistachio Growers, Joe and Carly, they're helping with enrolling growers and onboarding and training people. And then myself, Jalinda, and Chuck, we're going to be setting up and maintaining this regional network of smart traps. So, and we're all kind of doing a little bit of each other's tasks there as well, but just kind of a macro breakdown. So, what we're hoping to achieve here is more accurate monitoring of navel orangeworm as well as crop development. So the ability of navel orangeworm to infest the crop is mediated by the integrity of the hull. So we have these phenology models for the tree, so we want to overlay those with these pest forecast models for navel orangeworm to really give people an idea of where they may have greater or lesser risk of infestation. We're also trying to maximize the impact of mating disruption. This technology works better when it's scaled over large areas, so if we can aggregate groups of people together and and incentivize adoption. There's also the ability to better time pesticide treatments, looking at these regional trends with both tree phenology as well as navel orangeworm activity. We also are hoping to gain better insight on regional activity levels and crop damage in terms, the outcomes of these coordinated efforts. So if you're interested in participating in this, you can reach out to Joe and Carly. You can go by the APG booth. You can also, of course, email any of us on the project. But there will be more communication coming soon about this and more information sessions and training sessions to get people familiarized with this new platform as we roll it out. So again, want to acknowledge the support from the Pistachio Research Board as well as the Almond Board and CEFA to get this effort off the ground. And at that, I think I have a little bit of time left to answer any questions. So thanks so much.
All right. Hold it, Bob, in the back. And then, Bob, you're next.
I just want to say to the pistachio industry,
Oh, well.
we have fought navel orangeworm. I personally, back to the days of Dick Rice for 45 years. You must buy into this program. This is like a one-shot deal. I haven't spoken to Houston about this. I just heard this. We have been begging for this program for years. Houston is now providing you an opportunity to actually get a leg up on navel orangeworm. And as the CDFA gets tougher and tougher on pyrethroids, you're going to get your ass kicked before we solve navel orangeworm. So here's your chance. Thank you.
Thank you, Beatty.